Hi everyone, this is Ghalib and Substantia Nigra is with me. We are today going to continue our deconstruction of one of the videos that is very popular among the creationist circles online. And Stephen C. Meyer and those other gentlemen are seen as some people who are taking the challenges of evolution through natural selection head on, and they think that they are making some significant contributions against fight their in their fight against evolution, and they're trying to prove that evolution is not a fact. And we in the previous video deconstructed many of their claims, and we made it clear to you guys what were the fallacies that they were playing, and what were the illogical and non-factual things that they were saying. But today we are going to uh, take it further. Substantia is with me, and we are going to do that right now. So let's start it. I would just get it on my screen, and uh, hopefully today's agenda we are going to discuss. So Substantia, how are you doing?
I am very well, thank you. How are you, Ghalib?
Hello. I think I am unable to. Hello?
Can
Hello?
you hear me?
Yeah, now I can. I think my volume was somehow it got muted. I don't know why. So now I can hear you. Yeah. So how are you doing? And
I'm
how's
very
everything?
well. I'm very well. What about you?
I'm good. I'm good. So let me just bring this up, and where is this?
So while Ghalib brings up the slides, hello everyone, all the audience. I hope you are following these episodes. So in the last episode, as Ghalib was telling, we deconstructed some of the objections raised by those three gentlemen: David Gelernter, who is a computer scientist at Yale; David Berlinski, who is a mathematician and philosopher; and Stephen Meyer, who is a philosopher of science. They were talking about molecular biology, and eerily, there was no biologist on the panel. We mentioned that last time. And were they were actually working very hard and putting a lot of effort to make a case as if there are some mathematical challenges to the framework of evolutionary theory. And because Darwin at his time didn't know about molecular biology, so they were saying that Darwin's biggest problem was that he didn't know about molecular biology. So last time we actually uh, shared a lot of recent literature from molecular biology, which actually shows that some of the latest discoveries in the molecular biological mechanisms, they all make the case of evolution much stronger than it was at Darwin's time. So all these things, yes, they were not known to Darwin, but whatever has been shown, whatever has been discovered in the last like 40, 50 years, they have actually provided us with more fuel for evolution. So Ghalib would now probably go through the recap of what we covered in episode number 1 of this series, and then we will continue with this episode. So Ghalib, over to you.
So actually, we discussed their objections and the things that they were presenting as credible, and they were trying to portray themselves as that they were discussing whatever the cutting edge research that they were doing, and they were proving evolution wrong. So last time we discussed how they were wrong, because they raised a key objection, and that was that getting for a protein to mutate into a different protein was impossible. And let's say if a protein mutates into another protein, that would be just totally nonsense, and that would be a non-functional protein. So the overall function of life or the function of a cell would just break down. So because they were saying that proteins are unique and they have a very specific amino acid sequences, and a protein with a specific amino acid sequence can perform a specific job, and no other protein can perform the same job or behave in the same manner. So, so they were basically saying that the evolutionary process that science has explained to us is basically handicapped in producing new functionality when the protein changes, or if the protein changes, it cannot perform the duties of the protein that it was previously, according to the DNA sequence from which it has been encoded. So, we showed from the literature that there are multi domain proteins which have a very similar functionality despite having remarkably different amino acid sequences. And but the proteins that result from those sequences, which look very different, perform the very uh, same functionality. So
So
that
Ghalib,
is why we are saying that their argument or their basic premise was wrong. Yeah.
yeah. So if you want, just for refreshing everyone's memory, you can read one line from paper number 002. That's on multi-domain proteins. So that is what we showed
Let
for
me
every.
just pull that up. I think Acrobat is behaving very, it's not behaving very well today.
Yeah.
Let
So
me
one of
pull
the
it up from the other side. Just let me. So I'll pull up the paper one.
yeah. So
Just give me a
paper
little
number
bit
two.
of time.
Paper number two.
Number two, yeah. Okay.
So you pull it up. So
Hmm.
meanwhile, so guys, their main one of the key objections that was raised by David Gelernter and Stephen Meyer was that they were talking about that in order for a protein to function, we need exactly the same particular amino acid sequence. And if one amino acid changes, then suddenly the entire function of the protein would fail. This is what they were contending.
Okay.
Uh, this
It
speaks
won't
of,
behave in the same way. So I am going to try to close Acrobat because I opened.
we can actually see the correct paper.
No, but I would be able to. This is not Acrobat. I tried to upload it into StreamYard, and just give me a minute. I'm just pulling up paper number two.
Yeah, take your time. Take your time.
Yeah, no issues here. Now it will be fine. Okay, so now everything will be okay. And so I had to change a little bit. Okay, now it's okay. Okay, so now this is paper number two.
Yeah, just a little bit of in the second column, if you scroll down.
Yeah,
So
this here.
the green part, if we can just refresh that green part in everyone's memory in column number 2.
Yeah, so here it is. Yeah.
Yeah, they're saying that often structural similarities can be present even in proteins that share little sequence similarity, as structures more strongly conserved than sequence. So this is the hallmark of multi-domain proteins, where different parts of the proteins keep changing, but the overall function of the protein remain pretty much the same. So that contention of those three guys, that if you change even a single amino acid, then the entire function of the protein collapses and evolution cannot proceed. Their argument was hinged on this not even assumption. I don't know, wherever they were like imagining it from, they were hallucinating this so-called statement. This is actually not very advanced. This is a 101 of biochemistry. that the proteins, they share functional similarities, even though their underlying amino acid sequence is different. So their entire argument, their strongest argument, was based on this assumption. So we can dispose of that argument right away and call it a day, but we are going to continue having a deep dive into other aspects. So we can go back to the presentation, the slides, Ghalib.
Yeah, I'm just going back. So yeah, here we are. So their argument, as Substantia has told you guys, it was invalid. There was no substance in that, and it was just trying to be rigid about, or trying to just not accept the truth about evolution. And to be very frank, we also got an idea from the discussion those gentlemen were having, that basically they were not trying to understand how evolution happened and the basic mechanism behind evolution. So we explained a lot of sources of variation to you guys as well, and the sources of variation were important because they were trying to say that they were trying to portray as if mutations only happen through point mutations. Point mutation is the only type of mutation, which is clearly very wrong, and it cannot be accepted as a fact, because when you study the genomic processes that go behind the evolution of living organisms, you see there are so many mechanisms that can operate within the genome and change the genome drastically and produce new uh, traits in a living organism. So we talked about alternate splicing. We explained it to you last time. We talked about frame shift mutations, cumulative neutral variations, which are also known as, which are applicable in the case of pseudogenes. Then post transitional changes, epigenetics. We haven't touched it very deeply right now, but in the coming episodes, we are going to explain the methods of methylation, histone modifications, how non-coding RNA behave and operate within a cell of an organism. Then transposable elements, jumping genes, we are going to explain them to you in the coming episodes, and also about the heritable microbiome. This is that we slightly touched, but we are going to explain them in a lot of depth in the coming episodes. So you have to subscribe to this channel and keep watching out for the episodes that are going to come next. We
Yeah.
also yeah.
Yeah, so basically, we addressed all of their points because a common theme in all of their objections was that, you know what, uh, perhaps evolution can tinker with fine tuning and micro changes, but it cannot lead to large changes. Macro evolutionary changes cannot come about, and they can not be sudden. So we actually discussed all kinds of variations and all kinds of mechanisms and alternative mechanisms of inheritance, of inheriting variations across multiple generations, as Ghalib showed on the previous slide. So all those are sources of variations which can be sudden and which can lead to macroevolutionary changes. We so much so we talked about the epigenetic mechanisms of punctuated equilibrium being a situation which is the like a central theme in modern understanding of evolution, is that for very long periods of time in evolutionary history, there is very little change. There are periods of stasis or no change or very little change, and then there is sudden burst of variation and evolutionary like a flourishing of different species and sudden changes. So interestingly, there are epigenetic mechanisms which have been proposed to explain the phenomenon of punctuated equilibrium. There is something known as this epitransposon hypothesis. As I mentioned last time, this is referred to as hypothesis. Uh, it's their honesty of the uh, proponents of this hypothesis that they still choose to call it hypothesis, although it is backed by overwhelming evidence. So, if you are really interested, we had a deep dive into a very detailed paper which actually talks about punctuated equilibrium being the result of a tug of war between activities of transposable elements and epigenetics. So you can go back. We referred you to a book by Nelson Cabet on epigenetic mechanisms of Cambrian explosion. They talked about Cambrian explosion. There is work which proposes biochemical and molecular biological mechanisms based on epigenetics that can explain Cambrian explosion and sudden changes. And we concluded the last episode at this point that our modern understanding of molecular biology actually has unearthed a slew of mechanisms, an additional set of mechanisms, which can explain not only this gradual small steps of evolutionary change, but also fast, sudden, and macroevolutionary change as well. So this is developing into a consistent framework. And just to be very clear, people talk about and they criticize neo-Darwinism as if that's the established body or framework of evolutionary biology. The evolutionary biology's current framework for last many years, like almost like 3-4 decades, is not neo-Darwinism. It is basically something which is referred to as the uh, EES, that's called the extended evolutionary synthesis. That is what it is, in which there are multiple parallel mechanisms of inheritance. Yes, genetics being the main and central player in it, sure enough, but that is not the only source of variation. There are other sources of variation as well. And within genetics, point mutation is just a very, very small subset of the spectrum and the landscape of variations that are available for evolution to work on, as we saw the bulleted list on the last slide. So that was our ending point, concluding point at the end of episode number 1 in this series. So today's agenda, Ghalib, if you could go to the next slide.
Okay, so yeah, so this is today's agenda. Uh, Creationists say that there is a static mutation rate, and they also, on the basis of population genetics, which has been very popular in the past few decades, they try to tell you that mutation can only happen at a steady rate, and that's it. The rate of mutation or the rate of evolution or the rate of adaptation of different organisms according to their environments, it does not change. This is what people usually assume. And even in the scientific circles, this was what was assumed in the past, but people always had the idea that this was not the case. So we will dive deep today into the variable and accelerated mutation rates that scientists now have evidence for, and it has been proven actually. Specifically, we will discuss a review paper that was published in Proceedings of National Academy of Sciences, and this shows that mutation rates can significantly change when the MUT1 repair enzyme uh, it mutates or it changes in a way, and because of this, the supply of beneficial mutations also increases, and it accelerates the rate of evolution for an organism that is living in a specific environment. So this is what happened. So we will also present a mathematical challenge to the deniers of evolution in the form of George Price's result, the Price equation, the famous Price equation, which was basically given by George Price almost 50 years ago. And now people are starting to learn its new meaning. And that is what we are also going to explain here to you. And which also proves that evolution actually is mathematically inevitable. It's a thing that is going to happen, and it's as true as some of the other mathematical equations that explain our universe and our world. So, Substantia, would you like to add something more here, or should we move on to the next
Sure,
one?
sure. So you can bring up paper number one in parallel,
Mhm.
and let's set the stage for reviewing paper number one, which is on the analysis of mutation spectrum when the repair enzyme gene MUT1 is disabled and deleted. So we will probably go through the idea of repair enzymes first. So, but you can bring up paper number
Yeah, I am
one.
just going to bring it up. So, this is paper number one.
Yes, yes.
Mhm.
So the thing is this, one has to remember that when DNA replication takes place, and we talk about these things a lot during our Urdu streams as well, what happens is that we are told, we are told in textbooks that the DNA replication is a very accurate process. It's a very high fidelity replication process. And the error in the replication is of the order of 1 in 10 to the power 8 replications, like 1 in 10 to the power 8 base pairs. That is what the error is, which is, of course, remarkably high fidelity replication rate. But the important thing to understand is, as I always stress, that this high fidelity replication rate is not achieved in a single step. Actually, the replication error is 1 in about 10,000, which is actually a very big, significant error rate. What happens is that once the DNA has been replicated, there are dedicated mechanisms which are referred to as the error checking mechanisms or proofreading mechanisms, which make sure that the DNA base pairs have been copied correctly. And if an error is found, then it's a trial and error thing that there is a second attempt to copy, there's a third attempt to copy. So this high fidelity replication is actually a consequence of trial and error in and of itself. Okay, so that is what happens when DNA replicates. So there are many enzymes which are responsible for this DNA repair, and those enzymes are referred to as, of course, repair enzymes. MUT1 is one such enzyme. So if you can, uh, so one of the key results over here is this, that if you could go to that green part, which was highlighted, Ghalib.
Okay, this
Yeah.
here.
So they're saying that their experiments show that changing the mutation spectrum allows populations to sample previously undersampled mutational space, including beneficial mutations. So I'm going to give you the final result right away, which is as follows. What happens is that when the repair enzyme is working fine, so they have got a distribution of mutations. So there is a proportion of beneficial mutations even in those mutations as well, and a proportion is neutral, and a proportion of mutations is detrimental or harmful. What happens is when they mutate and delete the MUT1 gene, which is responsible for MUT1 enzyme, so the repair mechanism slows down. Okay. As a result, there are more mutations that are surviving in each replication process. So they are not being culled by the repair enzyme. As a result, the distribution and the kind of mutations also change. The number of mutations, the rate of mutation increases significantly, and the kind of mutations that survive now because of the lack of repairing process, that also changes. And as a result, what happens is the key result is that when the repair enzyme is not working, what happens is the supply of beneficial mutations go up. Okay, so we will have a deep dive into this paper one by one. But before that, I think that both Ghalib and myself thought that it's really important to convey to our audience the notion of randomness and the notion
Yeah.
of a spectrum of mutations and what kind of distribution of mutations should we expect. What is random in the context of mutation? Is it the case that the baseline mutations are extremely random? Is it the case that randomness is always bad? Is it the case that randomness is actually the fuel on which evolution feeds? Because the results of this study actually shows that randomness is responsible for beneficial mutations. So normally people have this preconceived notion as if randomness is always going to be bad and randomness is going to create harmful mutations. But the empirical results, what we are gathering now, this is a 2023 paper. Based on this study, the results, the understanding that is emerging is that randomness actually increases the proportion of beneficial mutations. So we will have a deep dive into this paper. But Ghalib, if you want to go back to your slides, we can like build some background in terms of what is randomness.
Yeah. So basically, Substantia, would you like to explain this, or should I start with
Okay,
it?
I'll explain. So this is very basic stuff, guys. So what I want to explain is that randomness in itself has a spectrum. Randomness in itself can have levels of randomness. Something can be extremely random, can be at the extreme end of randomness, and there are degrees of randomness in between. For instance, I think everybody is like familiar with rolling a die, a six-faced die. If it's a fair die, which means that it is not loaded, that actually means that every one face with every number is equally likely to happen. Okay, like you know that if it's not a fair or a biased die, then each one of those numbers is equally likely, and that probability is 1/6. So this is the extreme end of randomness. So when we everything is possible and the likelihood of every outcome is the same, everything is equally likely, that's the extreme end of randomness. Okay, so you just have to bear this in mind. If there is a little bit of different distribution as compared to what we are seeing over here, so here we are seeing something which we can refer to as a probability distribution. On the x-axis, you have the outcomes, 1, 2, 3, 4, 5, 6, and these bars, they are just representing heights, which are representing probability, and this is 1/6. Okay, if it was not totally random and a little bit biased, that would mean that some of the numbers could be more likely than other numbers. So all bars won't be of equal height if it were a biased distribution. A biased distribution means relatively less random as compared to this extremely random distribution. Now we will see, next slide, that what
Okay,
does
let me
extreme.
just explain them by an example. What do we mean by uh, that randomness? It's not an absolute randomness here that we are discussing. So let's say there were natural laws, and there are natural laws in our world. Like gravity is a natural thing that exists in our world. So if we would say that the world was totally random, then this would mean that a river which flows downstream would not flow downstream, and it was equally likely that it would start flowing upstream as well. So, in this way, nature is biased that rivers can only flow downstream, or rocks can only fall from the top of a mountain toward the bottom of a mountain. So, this means that the the natural laws that we have are not totally random. And like here we are saying, the if there's a six-sided die and you just throw it, so it's equally likely that there would be the one would be on the top or six would be on the top, because the die, if it's not loaded, as Substantia was saying, then any face can be on the top, and every face is equally likely to be on the top. So that would be absolute randomness. Yeah. So we go to the next stream. The
Yeah.
next.
So
Yeah.
we want to explain now what is absolute randomness in the context of mutations.
Yeah.
Okay. And you will appreciate, based on this example, you will be able to appreciate this within the next few minutes. So the four bases in DNA, the adenine, guanine, thymine, and cytosine in DNA, and of course, we have got a uracil as well, which is a base in RNA. So they can be grouped. So there are basically two groups of molecules, which is a larger class of molecules like purines and pyrimidines. Uh, so the adenine and guanine are the bases which come under the category of purines, and the cytosine and thymine, C and T, come under the family of pyrimidines. So one key difference which you can observe right away if you have just taken some basic high school organic chemistry, so you would be able to spot the differences between the structure. So one basic difference between the structure of a purine and a pyrimidine is that purine has got two rings and pyrimidine has got a single ring. So for our purposes, you can consider that A and G, adenine and guanine, belong to the same family of purines, and cytosine and thymine, C and T, they belong to the same family of pyrimidines. Next slide, Father. Yeah. So there are many kind of mutations that can happen from base to base. If you look at this chart over here, so you can see that if I am currently adenine, I can either become G, C, or T. Okay, and the same is case for the rest of the other bases. So if you think about it, how many possible mutations are there? So you can consider that, you know what, there are four sides of this square, and each side represents two mutations because you can move in either direction. So these are four sides and two mutations, so eight those. And then diagonally, you also have two lines, so there are four mutations diagonally as well in this square. So 8 plus 4 is a total of 12 possible mutations. Let's see what kind of mutations there are. So if we have a mutation within the same family of molecules, that mutation is referred to as transition. For instance, A to G and G to A is purine to purine. So Ghalib has shown that in blues, that's a transition. C to T and T to C is also within the same family of pyrimidines. That's called a transition. Any mutation which is across family from purine to a pyrimidine or a pyrimidine to a purine, that's called transversion. So red arrows represent transversions and blue arrows represent transitions. We
So
will,
these are the two types of mutations, two types of point mutations. Actually, one nucleotide base is changing into a different nucleotide base. If it changes into the same type of nucleotide base, if a pyrimidine changes into a pyrimidine, it's a transition. If a purine changes into a purine, it's a transition. If a purine changes into a pyrimidine, it's a transversion. Or vice versa, if a purine changes into a pyrimidine, then it's also a transversion. So this is what we are trying to make you understand here, and you can see that these are the four alphabets of uh, our nucleotide alphabets that are basically the very definitions of the recipe of life, and all of these can change into the other alphabet, but the changes are basically different, and their rate also varies, that Substantia is going to explain to you further. So yeah, should we move on?
yeah, next slide.
Okay, so yeah.
So let's consider what is extreme randomness in the context of these mutations. Because if you have seen, if you have spotted some pattern among creationists and deniers of evolution, they always frame the discussion by considering that, you know what, this is one upon the number of possibilities, each one is one upon the number of possibilities. So whenever you are trying to compute a probability by saying that the probability of any particular outcome is one upon the number of possibilities, that actually implies as if you are assuming that the case corresponds to absolute randomness, because only in the case of absolute randomness do we have a situation where every outcome is equally likely, and therefore the probability of every outcome is 1 upon the number of possibilities. But we will see whether that's the case empirically or not, and why the computation of probabilities of the creationists and deniers is always wrong with probability 1. And of course, the pun is intended over here. So, in the case of these mutations from one nucleotide base to another nucleotide base, so as we saw on the last slide, there are 12 possibilities. So, along the x axis, we have got those 12 possibilities. And these orange or gold bars, they are representing those mutations in which you have either a purine to purine or a pyrimidine to pyrimidine change. So these are transitions. And the blue bars represent transversions. They are across family. So a total of 12 possibilities. If we were on the extreme end of randomness, which means that every mutation was equally likely, if things were indeed absolutely random, as many creationists and deniers of evolution think, because they have never gone to a molecular biology lab and they've never experimented and they've never computed the rates of mutation. They, sitting in their armchair, they think, you know what, there could be 12 possible mutations, then the probability of each mutation is 1 upon 12. So that would be the distribution of the probability had the mutations, uh, if the mutations were absolutely random, then this would be the situation of the probability distribution. But we will see what it actually means uh, in terms of actual mutations in an actual living organism. But let's try to analyze how this thing can also be represented in terms of the distribution of transitions and transversions. If you think about it, in this absolutely random case, it seems like it's obvious that there are more ways to have a transversion. There are more blue bars than there are orange bars or gold bars. So there are more transversions as compared to there are transitions. There are only 4 transitions, and then there are 8 transversions. So the number of transversions is twice as compared to the number of transitions. So if we plot a probability distribution of transitions versus transversions in an absolutely unbiased and absolutely random case. By the way, when we are at the extreme end of randomness, we would also refer to that as the unbiased case. Okay? So if there was no bias involved in mutations, and we were at this extreme end of randomness where every mutation was equally likely, in that case, transversions would have been twice as likely as compared to transitions, which means that in a pool of mutations, if this was the case, you would expect to see 2/3 of the mutations to be transversions and 1/3 of the mutations to be transitions. So, Ghalib, you can go to the next slide.
Okay. Yeah.
Yes. So this is the situation of transitions versus transversions. So now you can consider that we have compressed those 4 bars into 1. So you have a 1/3 probability, and those 8 bars into 1 blue. So you have 2/3 probability. And in terms of transitions versus transversions, if we were totally unbiased, then that would imply that 2/3 are going to be transversions and 1/3 is going to be transition. But let's see if that's the case or not. Okay, we will see that.
Should we move to the next one?
Yeah.
Okay.
It turns out that this ratio, which is in the heading of this slide, the caption of this slide, we see this TS by TV. That's called the transition to transversion ratio. This ratio is a very handy number, which has been like observed and measured and computed for various species in multiple organisms. And more often than not, what happens is that we have a situation which is not equally likely situation. It's actually a situation which can be considered as an extremely biased situation. the leftmost thing could be a situation in the case of E. coli. So you see this wild type word over here. Wild type actually means something that occurs naturally, which hasn't been artificially mutated in the lab. So you would come across these terms, wild type, and a short for it is WT, and or a mutated version or a mutator version, as opposed to the wild type when we are trying to study various types. So the leftmost situation, again, TS means transition, TV means transversion. We know if the situation was unbiased, which is the third plot on this screen, that actually means that we have a blue bar, which is 2/3, and an orange bar, which is 1/3. But if I reverse this thing, which is the leftmost first plot, that is actually an extremely biased situation. And in E. coli, which is a test organism over here, in that organism, this ratio of transition to transversion can actually vary from 2:1 to 4:1, like extremely biased. Where we know that unbiased actually means 1:2 because it's transitions to transversions. So the unbiased is the third plot. In between that unbiased and this very biased situation, you would come across a relatively less biased, which is the second plot. It's very important to remember that 50-50 in this case is not unbiased, because unbiased, which is absolutely random, just because by the virtue of transversions being twice as many as transitions, unbiased means that transversions should be twice as many as transitions. So 50-50 is actually not totally unbiased. It is less biased as compared to what we have on the leftmost of this slide, but it is more biased as compared to the unbiased case. And you can also have a situation which is referred to as the bias reversal, which means whatever bias existed in the wild type, if you can somehow reverse it, that's the other end of the extreme, which we see on the rightmost plot over here. So what we are going to discuss with you through that paper, by reviewing that paper, is that both empirical results and simulation results, they point to similar results. And what are those results? They show that reducing the bias or reversing it is actually beneficial. And when MUT1 gene is deleted, you either reduce the bias, which means you become more random, or you become biased on the other side. Either way, with MUT1 gene deleted, beneficial mutations increase. Okay, which actually means that whenever there are situations when there is mutation, there is one mutation. So it's very important to understand the downstream effect of certain mutations. Because MUT1 gene in itself is responsible for the repair enzyme, if there is a single mutation, a single point mutation that occurs in that MUT1 gene, that single mutation would basically trigger a domino effect of mutations elsewhere as well, because now the DNA replication process has lost its watchdog, so to say. So just one mutation can actually significantly, non-trivially, significantly increase the rate of mutation, and also it can increase the type of mutations that are now being explored. As a result, what we learn from this basic understanding of molecular biology is that small changes can actually lead to large consequences. If you have one small change in MUT1 kind, that's going to be a giant leap for the DNA kind. Okay, so this is exactly what it means, and we
What
can now, yeah,
Substantia, should we also explain what actually MUT1 does? So
yes,
what actually MUT1 does is it is biased. It is biased towards transitions because it will somehow overlook the transitions. But whenever there is a transversion, it would correct it. So, so the MUT1 enzyme basically selects the, in a way, selects the mutations that it allows, and it never it there are less transversions that it lets them pass through into the written code of DNA. So when a mutation happens in it, then it changes and it starts to allow transversions to increase. And eventually, Substantia is going to explain ahead how that affects evolution of that organism on a larger scale. So that is
By the
what
way,
we are trying to explain.
Ghalib, I'm glad that you brought this up. That Matwai is biased. I
Yeah,
really
right.
like that interpretation, and there is no magic behind that as well. If you think about it, it is as you said, it is forgiving to transitions, and it is not forgiving
To transfer.
to transversions. The and this is also not magic, because if you think about it, detecting a change across the family transversion is easier.
Easy. Yeah.
Like transition is a within
Because it's a
family
big
change.
change. It, the
Yes,
one circle is changing into two. So it's a
and
big change. Yeah.
Matwai in itself also is not super perfect. Actually, it's very difficult for Matwai to detect a transition. That's why transition just slips through, because it's a single ring. You have got one ring to one ring, or a two ring to two ring uh, variation. So it's very difficult to detect those small changes. Whereas if you have one ring to two ring, or a two ring to one ring conversion, it's easier for the enzyme to detect that. So that's also not very magical. That why that bias exists. That's just a consequence
Yeah.
of the fact that you are making a jump within the family, within of molecules, or across the family of molecules. So it's very simple. So
So one thing I also want to make clear here is that when I talked about a river flowing downstream rather than flowing upstream, it's it's the same case here as well. Whatever is more likely according to the laws of nature, that is basically more likely in the case of evolution. So there is nothing about life that is, as many creationists try to explain, that life is totally anti nature. Life is growing against the laws of nature. No, it's not like that. Life is actually progressing and evolving in alignment with the laws of nature. There's nothing going on that is contrary to the laws of nature. Even when the law of this enzyme, that the law that this enzyme sets in our genome, when even that breaks, it also produces an outcome that is extremely likely. There is nothing like something happening here that's not likely. All of this is natural, and all of this is flowing downstream like a river flows from the top of the mountains towards the Great Plains and towards the sea eventually. So all of this is natural. This is not something that is unnatural. Yeah.
yeah, so we can go back to the Matwai paper again, and then read through
Yeah.
some of the key passages of the paper and see the results.
Mhm.
Okay, so again, the title of the paper says it all. If you can read between the line, that what it is saying is shifts in mutation spectra enhance access to beneficial mutations. So before the MUT1 gene was deleted and tinkered with, the mutation spectrum was something else. There were more transitions and less transversions. And there was this proportion of beneficial mutations and harmful mutations and neutral mutations. What they did was they tinkered with the MUT1 gene, and as a result, the mutation spectrum has shifted. And as a result, those mutations which were not being explored, those parts of the mutational landscape which were not being explored or undersampled before, now start getting sampled. As a result, the proportion of beneficial mutations goes up. When you start doing more experiments, albeit randomly, of course, you increase the supplies. Those things which were not being considered as candidate mutations before, now they belong to the pool of candidates. It's as simple as that, but we will read some of the stuff over here. Ghalib, if you could scroll down. I think if we read out some part, the initial part for our viewers, it would be easy. I'll just read this part from actually start of the first paragraph. If you could scroll up a little
Okay,
bit.
here.
Yeah, so I'll start. The mutation spectrum, yeah, the mutation spectrum describes the frequency of various classes of sampled mutations. Okay, for example, transversions, transitions, and is often governed by the action of DNA repair enzymes. This is a key statement. I think we have built the background so that you can understand this very first sentence, that mutation spectrum is just the distribution of the different kinds of mutations, and it is often governed by what? It is governed by DNA repair enzymes. How accurately or relaxed the DNA repair enzymes are, they are the key players behind the shifting of mutational spectrum. First of all, this is a clear cut answer to all those people, all those calculations who perform those detailed calculations of waiting time problem and all those things. They select one particular mutation rate. They do not bring into their models this aspect of changes in the repair enzymes and the changes in the mutation spectrum. They're just totally ignoring. They are actually situated in 20th century old molecular biology where they had no idea that the mutation rate in and of itself is such a variable quantity. Okay, let's read the next sentence. What are they saying? They're saying by determining the pool of genetic variants available for selection, mutation spectra can shape key genome features. For example, nucleotide, codon, and amino acid composition, even if under selection for thousands of generations. Determine the genetic basis of adaptation, driving convergent evolution, and shape the evolution of resistance to antibiotics. So, why is it the case that repeatedly we see similar structures evolving in evolutionary history? That is called convergent evolution. When similar kinds of structures keep evolving over and over again, for instance, one of the most celebrated examples is the evolution of I. It has probably evolved more than 36 times in evolutionary history, like independently evolved, not by descent. Of course, there is going to be correlation of phenotypes when you are actually evolving by having a common descent. But some of the features, it can be shown, have evolved independently of each other, but those features are very similar. And they are examples of what is referred to as convergent evolution. Evolution repeatedly converges to a solution. Why? Because there are certain biases in the mutation spectrum. Okay, Ghalib, if you could just hide this menu that is
Yeah.
like occluded. Yeah. So the mutation spectra are important for adaptive evolution, but their role is underappreciated. Okay, so this paper is actually last year paper, 2023. So as of 2023, the authors of this paper, the researchers, are actually truthfully stating that even today, the understanding of mutation spectrum is not very well appreciated. It's underappreciated, although this is one of the key players and the driving forces behind evolution. More importantly, what is highlighted in yellow over here is that the mutation spectrum is instantaneously and relatively easily altered by loss of function mutations in DNA repair genes, as is observed in mutators, and also that can have higher mutation rates. So as a this can happen easily. You don't necessarily have to tinker with this enzyme. Of course, for the purposes of this study, they will be tinkering with this MUT1 repair enzyme themselves. But this happens all the time. That there are mutations in MUT1 gene and other repair enzyme. MUT1 is just one kind of repair enzyme, and there are other kinds of repair enzymes as well. But what they're saying is that in nature, this mutation spectrum is instantaneously and very easily altered. So it happens very easily. Why? Because it requires very little amount of variation. Even one mutation is enough and can happen simultaneously, which means that within a matter of few generations, large changes can happen. Okay.
Deficiencies and strong selection pressures can lead to mutations in MAT2 gene and other repair enzymes. So
Absolutely, absolutely.
that's also very well referenced and documented by this team, and at and many other research papers have discussed that, uh, you know at length.
Yes, yes. And of course, in the next line, they're saying this elevated mutation rate, the sentence in blue, the elevated mutation rate increases the supply of beneficial mutations, facilitating rapid adaptation. And as Ghalib was saying, that these spectral shifts can also occur with the loss of bacterial DNA repair enzymes and under nutritional and other kinds of like stress, like anaerobic stress. So your environment changes and your mutation rate can change and can become faster. Okay, so you can become a high speed mutator in response to environmental stress and nutritional deficiencies. Okay, so let's scroll down.
And especially Substantia, also we need to clarify this here as well,
Mhm.
that people think that if a, an organism is changing at a very rapid pace, then it might become something like a mutant that they imagine in the movies. That's also not the case. That is what we are going to explain further, and Substantia is going to dive deep into this. Yeah.
Yeah, yeah. So first of all, those mechanisms that uh, we discussed last time, they kind of answer those kind of questions as well. First of all, if there are protein changes caused by these mutations, suddenly the proteins cannot change very much. Okay, and if there are pseudogenes, which means that they are neutral variations, they are copies of the genes, they can keep accumulating the changes at a very fast pace, but because they are pseudogenes, they are not being transcribed right now, so they're not directly under selection. However, there are parts of genome which is actually under significant selection as well. And sure enough, if they are not beneficial at that rate, if the mutation rate increases and it is unable to create a stable supply of beneficial mutations, sure enough, those mutants will get extinct. However, we should be asking this question: is it empirically the case? Does empirical data really suggest that when we increase the mutation rate, then we create mutants which are not beneficial for that particular species? That is not the case what actually lab studies are telling us. Okay, it's one thing to declare something by sitting in an armchair, and it's a totally different thing doing an actual experiment. So this is what they have done. So what they did was this pink part, we manipulated the mutation spectrum by deleting a DNA repair gene, creating delta MUT-Y. So the delta MUT-Y, they are calling it that strain, which is the mutator strain. From what? From the wild type strain, the naturally occurring strain. Okay. And as a consequence, they change both the mutation spectrum and the rate of mutation as well. And then they allowed both the wild type and the mutator version to evolve independently. So the next line says that allowing independent lineages of both strains to evolve under mutation accumulation, we sequence whole genomes of evolved isolates to identify strains carrying a single new mutation each. Interestingly, when you will tinker with MUT-Y or even baseline mutation rate, you will have mutations which are single point mutations, 2 point mutations, 3 point mutations, so on and so forth. They said, you know what, let's throw away non-point mutations. Even within the scope of point mutations, let's analyze what is going on. So the so the actual variation is far more than they have considered in these experiments. And even in these limited, when they are constraining the variation by only considering single point mutation, even then the supply of beneficial mutation goes up we will see. And they are saying that they are measuring the effect of each single mutations on population growth rate. So the population growth rate of each strain is being considered as a marker of fitness. Of course, whatever strain produces more copies of it genetically and evolutionarily, that is the measure of fitness. How much descendants that you leave before you die, the that's the measure of fitness. So if you scroll down, Ghalib,
Yeah.
scroll down.
Should I go to this
Yeah,
figure?
this part is
Okay.
this part. Yeah.
Results and
So
discussion. Yeah.
the main result is that the mutator has more beneficial mutations. Okay, and they measured the selective fitness effect, which is referred to as the relative growth environment. Actually, they did this in multiple environments, and in every environment, and by environment means if it's a like a organism, which medium the organism is contained in, and then they have got environment specific like fitness functions. Uh, and they compare both the mutated strain, the MUT1, and the wild type. And the chart on the right side of the of this page, this is in a nutshell their key results. And let me summarize it as follows. So what they're stating is, by the way, these labels on the left-hand side, these are uh, environments. The medium, for example, maltose, lactose, fructose, that's the environment in which they were growing the culture. And if you look at the top of this particular figure, you have got mutator, so mutator and wild type. So let's first read the numbers corresponding to wild type. So in wild type, when the MUT1 gene, MUT1 enzyme was still working and it was favoring transitions and culling transversions, what was the distribution of the mutations? 39% were harmful mutations. Sure enough, large percentage, 39% deleterious mutation in wild type. Neutral means they were neither harmful nor beneficial, 33%. And 26%, or is it 28? 28% beneficial mutations. By the way, even in wild type, beneficial mutations were significant, even when there was this baseline mutation rate, which was slow. Even in that slow mutation rate, 28% of mutations turn out to be beneficial. More importantly, you have to realize that selection would weed out only 39% of the mutations. The neutral mutation would still.
I think I should zoom in on this actually here,
Yes, yes, yeah, please.
because people wouldn't be able to
Yeah,
understand what
because
we're
I am
talking about
reading
here.
from these
Yeah.
numbers and the
So here,
text size
here,
is very small.
yeah.
So if we just look at wild type, WT first, mutator we will analyze in a minute, but just focus on WT. Even there, about 39%, let's round it up to 40%. About 40% mutations are harmful, but 60% mutations are either neutral or beneficial. So evolution, the selection would weed out the 40% of the mutations. Selection would not weed out the neutral mutations. Of course, selection would favor the beneficial mutation, sure enough, but it will also not select out or weed out neutral mutations. So the neutral mutations can keep accumulating from generation to generation, from generation to generation. And
So what
till
would be Substantia? Can I just simplify for the people here?
hmm,
Basically, MUT1 is like a watchdog, a policeman. Maybe it's the police of the genome. So if, let's say, a part of genome changes, so what it does is it does not let it change. And when it is present, so more crime is happening in a way that more harmful mutations are happening. And when you just remove the police or the MUT1 gene, or you change the police in a way that it is not that much functional anymore, so it's what is happening is just counterintuitive. And especially to the people who are creationists, they would think, okay, if more mutations are happening, then, and especially Michael Behe and his lot that we are basically discussing here about, they think Michael Behe's whole claim is that whenever there is a mutation, it is always loss of information in the genome, and that basically results in loss of functionality eventually. And because of loss of functionality, the change that happens is what people, according to him, what people wrongly term as evolution. But here, what we are seeing is when MUT is not properly functional, what is happening is more beneficial mutations are happening. And if you see here at the mutator type, you would see that deleterious mutations are
They have gone
decreasing.
down. They have gone
They're
down.
going down,
Yeah.
and beneficial have gone up.
Both
And neutral.
neutral and beneficial mutations both have gone up.
Yeah.
And
So
taking
this
a leaf
yeah.
and Ghalib, taking a leaf out of your example of watchdog and a policeman, you might want to think that that policing is enforcing conformity
Yeah.
and is discouraging novelty.
Yeah.
Okay, so anything that changes significantly just culls it down. So basically, it is trying to be very conservative. It's very conservative policing that is going on. It is enforcing conformity. It is against change. It is discouraging novelty and
Yeah.
innovation. When MUT is relaxed or is deleted, what happens is that you have got more novelty and innovation injected into the pool of mutations. As a result, those mutations which were not being explored before under that policed environment, now they are being explored. And as a result, you have got novel and innovative solutions in the mutation spectrum. Therefore, the beneficial mutation also goes up and the neutral mutation also goes up. So this is a remarkable result, guys, that by moving away from that biased mutation spectrum, by increasing the mutation rate when the MUT gene is compromised. And this is just one particular example. MUT is just one repair enzyme. And there is a single mutation, or you just delete one repair enzyme, suddenly you start exploring the mutation landscape such that the supply of beneficial mutation goes up. So this is remarkable, and it's just I think,
It makes a lot of sense.
yeah,
Sorry, just one thing I'll like to add here. So we've always seen throughout the fossil record, and also after huge changes in the environment, as close as within the last a, a few 100,000 years, we've seen this. After a glacial episode in the on the face of Earth passes away, there is a rapid, outburst of organisms evolving at a new organism that we see in the fossil record. And why do we see them there? We always see them there because that is a time of rapid environment change. So after every mass extinction, all six of them that have happened that we know of on this planet, we see rise of new organisms. This is how mammals took over. This is how even the dinosaurs took over from the reptiles. And this is how reptiles took over from the insects, because, at one point, insects were ruling this world, and then the reptiles came over. So, this is what has happened after every mass extinction on this planet. And this is how it makes sense. Because whenever life goes through such a period where the environment drastically changes and there are new selection pressures, and now we know empirically that at that time there are mutations in the MUT gene, and we also know that this is not a gene that is a new gene. This is one of those very old genes that might have existed since the time of, as Substantia was talking, uh, since the time of Cambrian explosion, even
so
more.
this is of course E. coli, one
Yeah.
of the very basic organisms. Actually, Ghalib, I'm again happy that you brought this up, because anybody who is hell bent in terms of finding faults and trying, like, some kind of to find some kind of loophole in evolutionary evidence might ask us that, okay, for E. coli, you have got MUT5 gene. What about other animals? What about humans?
Yeah.
Okay, I would I would request you to open this link. I've just sent you this link at the UniProt, which is the protein database.
Yeah, and we will show you that it is in you and me.
Yes, yes, I want to show that if somebody asks you that, you know what, MUT5 is an E. coli, and we would show you, we would demonstrate that some
Yeah.
other
Go ahead.
folks also have MUT5 gene. If you go top, what's the name? And that gene is referred to as the corresponding protein is MUT5H,
I
and H
H. Yeah.
for human or Homo sapiens. So MUT5H, we have got MUT5, and what is the function? So again, guys, this is a like legit source. You can go and search a UniProt database yourself. So you look at the URL on top in the address bar of Ghalib's computer. This is uniprot.org, like a reference database of proteins and genes. So MUT5H, organism, Homo sapiens. What's the function? If we read the function, if you could highlight the sentence under the heading of function. Yes. Involved in oxidative DNA damage repair. So not
And what
only
happens when it mutates in human beings?
exactly the same
Yeah.
thing.
That's
Exactly the
also
same.
that will also explain to you like what we are talking about here.
Yes. So not only is it the case that we have got MUT5 gene, it is functional. It is not a sleeping gene. Okay, it's a functional gene in humans. And what is the function? The function is also DNA damage repair. So if you have got a mutation in MUT5 gene in humans, a single mutation, what you get is an accelerated mutation rate, even in
And what
humans.
happens to such individuals who have this mutation in human MAT2 gene? A very high incidence rate for cancer that happens in human beings. And we know why we have cancer, because mutations are the reason why an individual gets cancer. So different types of cancers just spring up in those human beings who have this. So we can see that that's okay. Yeah. So that's what happens in human beings as well.
Yeah, and it's the point is that humans are also evolving. We know we have seen that we have seen populations of humans evolving over time, especially those underwater divers where their spleen size had changed. So when the environment changes, the rate of mutation increases. So even in humans, they can actually create an accelerated rate of mutations, creating a landscape of mutations where possibly you can have beneficial mutations. The likelihood of hitting beneficial mutations actually goes up. Okay, so that's the thing that
But the
it,
harmful mutations also could be there, but beneficial mutations
yeah,
would
yeah,
outweigh them.
just
Yeah.
just
Yeah.
like you have got more experiments, when it's as simple as that. When you start experimenting more, you can have harmful effects, but you also increase the likelihood of hitting beneficial mutations as well. And if you look at a simpler organism like E. coli, you have got experimental evidence that the supply of beneficial mutations actually goes up, and the supply of neutral mutation also goes up. So I'm stressing neutral mutations over and over again because neutral mutations also have non-trivial and extremely important role in evolution, in long-term evolution. Because they are neutral, I will repeat, selection is not acting on them immediately. As a result, they can keep accumulating more and more changes till the time they become either deleterious or beneficial. But as long as they are neutral, selection is not acting on them. So variation can keep accumulating, and then one more mutation can make them beneficial or otherwise. So that's really important that large changes can occur through those neutral mutations as well. And again, biochemically and from a molecular biological point of view, this seems very clear. Of course, we do not believe in any kind of prophethood, you know that. However, this idea of neutral mutations, if you go back to chapter 4 of Darwin's Origin of Species, you will find that idea of neutral variations as well. And exactly in so many words, that neutral variations could become a fuel of long-term evolution. This is what Darwin wrote, that because selection doesn't act on them immediately, they can keep accumulating till the time they become consequential. Right? So that's the case about neutral mutations. So we can come back to our slides.
Okay, so here we are back to the slides. Okay, should I move on? Should we explain a little bit here? So
Yeah, we spent a lot of time on this slide actually.
okay,
Yeah,
okay. So I think this was the last one.
Okay, very well.
This was the last one.
So,
yeah.
so this actually completes the story of MUT gene. We wanted to have a deep dive into it, and we have demonstrated to you one of remarkable studies that was published in the Proceedings of National Academy of Sciences last year. And we have just seen that what mutation spectrum is, what mutation spectra are. MUT is only one kind of repair enzyme involved in ensuring the high fidelity replication of DNA. And we see when there is a mutation or a deletion in MUT gene, what happens, mutation rate goes up, and as a result, the rate of mutation goes up, and also the proportion and the supply of beneficial mutations goes up. So I'm not sure how we are doing with time. If we have time,
I think we have 15 more minutes.
Yeah, time permitting, let's keep up. Let's keep our promise, and let's talk about that mathematical challenge that we want to present.
I think we should move on to that, and maybe if something is left, we are going to discuss it next time, because we are going to, I think at least one more episode is going to be on this same topic, then we are going to touch something else.
If you could open paper number 9.
Okay. Just give me 1 minute here.
So guys, George Price was a statistically oriented mathematician, a remarkable mathematician of the ranks of like those giants like Ronald Fisher. So George Price presented his Price equation in a short paper in 1970. And of course, when these models are presented, initially people think that they are very theoretical till someone comes and tries to run some simulations and tries to match some empirical data. And then these equations actually go into background. And we know that unfortunately, the training of many people is not that multidisciplinary, and that's just the nature of the beast. Once you specialize, you diverge along your own lines of specialties, and people in working in biological circles like lose track of mathematical details. But I think there is now increasing interdisciplinarity in the field where people from multiple backgrounds are collaborating with each other, and it's such an exciting time. So this is a paper from a special issue. Special issue is occasionally journals declare that in, let's say, in the month of December, coming month of December, we will have a special issue dedicated to a theme or dedicated to a topic, and they invite like participation and material and paper and research articles from researchers all across the globe to like from those topics that conform with the theme of that special issue. So if you read the title of this particular paper, the title of this paper, and this is from 2020, the title of this paper is Price's Equation Made Clear. And if you read the last sentence of the abstract, the grayed out part, the last sentence of the abstract, Ghalib.
This here? Okay,
just the last sentence. So
okay. The article is part of theme issue 50 years of Price equation. Yeah.
So the
Which year?
special issue was precisely to celebrate 50 years of this equation,
Price
price
equation.
equation.
Yeah.
Okay. So I'll just show you what this equation is. Don't get scared. Don't get intimidated. I will interpret each and every term in it or every symbol in it. It's not very complicated. And then I'll read some text out of this paper. So scroll down, Ghalib, to page number 3 of this paper.
Yeah, this is the price equation.
Yeah, you can scroll down because there is also, this is repeated down as well
Mm,
on the next page.
yeah. Should I zoom
Okay.
in a little?
Yeah, if you zoom in. So this,
Okay.
yeah. Okay, good. So this is the price equation, guys. Don't get intimidated. I'm not going through the proof. If you're interested, I'll be happy to discuss the proof with anybody offline. Okay, it's not very difficult. If you have got good mathematical background, you will get it. However, let's try to interpret what it is saying. So Zi is any particular trait uh, belonging to the ith population member. That's about it. Zi is any trait. It's a symbol for any trait. It could be height of something. It could be the production of insulin. It could be the production of a hormone, production of a receptor. It could be the color of your eyes. It's general. So this model is so general and so powerful. It doesn't matter what the mechanism of heredity is. It doesn't matter whether genes are the unit of selection. It doesn't matter whether inheritance is happening through genes or epigenetics. You think of any trait. So Zi is any trait. So the left side is, if you read the left side of this equation, it says delta E of Zi. It says change in the expected value of a trait between generations. Change in the expected value of any trait between generations. So that's the left-hand side of the price equation. On the right-hand side, I'm actually reading the yellow part, Khalid.
Okay, this one.
Yeah,
Okay,
yeah.
you are reading this.
No. The
This
yellow,
one. Yeah, yeah.
the first yellow part, Khalid.
Uh,
Yeah, you have got green and then 2 lines of text, and then you have got an equation in yellow, right? On top.
yeah, yeah. You are reading this one.
Yes.
Yeah.
Because this is the basic equation. Okay. I'm just going to quickly interpret this. So left-hand side, I've already interpreted, okay, which is the change in the expected value of a particular trait. Zi is a trait. W is a symbol which is being used to associate the fitness of a particular trait, and that would mean that in terms of ratio, how many offsprings that you leave. Any number that tells that, okay, this is more fitter than the other one. If, let's say, the W1 is associated with a more fitter trait as compared to W2, then W1 would be greater than W2. It would have more fitness. So if you see the first term, that is the product of the trait value itself, Zi, and its fitness. So what it's saying is the first term is the expected value of the product of the fitness and the trait value. Then the second term with a minus sign is the product of the expected values of, and expected value is just the average value in a population. So expected value is another term for average. So this is the expected value of the fitness times the expected value of Z. So E you can consider as just a symbol for expectation. And then on the right-hand side, you have got expected value of the fitness time, the change in the trait value. What you can show is that the first 2 terms can be simplified as something known as covariance between 2 variables. If you know a little bit of statistics, that actually means covariance actually means how the 2 variables change in relation to each other. For example, if something, it's similar to correlation,
Temperature and pressure would be a good example.
it's similar to correlation, but not exactly correlation. But if 2 things always change together in the same direction, if one thing increases and the other thing also increases, you will have a positive covariance between
Covariance.
them.
Yeah.
So this is the covariance of W and Z, which means this is a covariance between the fitness of the trait and the trait value itself. And then the second term I've already covered. So what it expresses, if you read this yellow part, is trying to interpret it. Of course, Price interpreted it, but people just ignored it. Okay, so the first sentence over here is this, that Price's equation expresses overall evolutionary change as the sum of a selection term. So this first part, this covariance, is called the selection term, and the second part is called the transmission term. And you will see why the first part especially is called the selection term, because it is the, the variation, the covariance of the fitness and the trait value. You will see what is going to happen on the next page. On the next page, let's let's skip the mathematical details. Let's skip to the result because, of course, okay. So let's read this. So what this 2020 paper, which is interpreting and revisiting Price's equation, what they're concluding is that, in other words, Price's equation represents a return to the original Darwinian logic of natural selection. Okay, so people say that Darwinism is dead. Actually, mathematics is showing us, mathematics is announcing the return of Darwinian logic. Those guys, David Gelernter, David Berlinski, in their relaxed armchairs, they were like talking about like high school level, like preliminary mathematical understanding of probability, which was totally in isolation from the understanding of molecular biological problems. So they were just making things up. So what the actual mathematics is saying, it's saying, and it's not like a simple mathematics like 1 upon 20 something. This is an actual proof of a theorem. Okay, which, so what is it saying? In other words, Price's equation represents a return to the original Darwinian logic of natural selection. And this was perhaps best demonstrated by Price. Like Price demonstrated this, but people ignored it when he showed that the selection covariance, so the covariance term, can be expressed as the product. I'm sorry, it's going to be a little bit technical, but let me read and I'll explain. Can be expressed as the product of the least squares linear regression of fitness against character value and the variance in character value. So what Price showed in a few lines of proof in 1970 was that this covariance between the fitness and its value can be broken down into this product. And the product on the right hand side of equation 4.1, this first part beta. So if you have seen any data distributed, a scatter plot, and if you know that you fit a regression line to it, okay, so of course that line has got a slope and you've got the regression coefficients associated with it. So if you fit a line between the fitness and the value of the trait, that is what beta is. That actually means that as long as there is any correlation or connection between that value of a trait and its fitness, you will have higher beta values. Variance in Z, as long as there is variation, some like variance is just like the spread in the distribution of any quantity. And this 4.1 actually captures, I'm reading it from the paper, blue part after green, as this captures Darwin's basic argument that selection occurs when there is variation in a character of interest. Whenever there is variation in any character, selection would occur, which means that you have got a non-zero variation that is correlated with the fitness, which means that this beta is also non-zero. Then you will have change in selective pressure. So this is a mathematical inevitability that there is going to be change in trait as long from generation to generation, as long as certain conditions are met. What was Darwin's original logic? Anybody who has read Origin of Species, Darwin's original logic was that, first of all, there should be variation in the population. Okay? And he said that cannot be denied. This can be observed that with respect to various traits, we see that there is variation in organism. That was part one. Part two, that different organisms with different values of that particular trait should have different fitness, which is the condition of differential fitness. Okay, which means that you have got differential fitness, which is this beta term. So Darwin's second line is now appearing as a term in this theorem. First line is appearing as the second term, and the second line of Darwin's logic is appearing as the third term. And last part was what Darwin said, that as long as offspring is more closer to their parents, of course, this means that inheritance is going to happen. Then that's the third part. That's the transmission term that was defined on the previous page. So Price's equation, without actually considering any excerpt from Origin of Species, is derived from first principles and outcomes Darwinian logic as encoded in this mathematical theorem known as the Price's equation. Okay, so by the way, any mathematically inclined person, again, if Dr. Gelernter is watching this, I'm sure he's aware of Price's equation, but I would recommend him to reconsider and reevaluate the underpinnings of the Price's equation and how that perfectly aligns with Darwin's original logic. Forget about the modes and mechanisms of inheritance. What happens now is this is independent of the mode of inheritance and the mode of transmission. And at the time, they were thinking solely in terms of point mutations, and they were restricting themselves only to genetic change. Now we see that within the realm of genetics, we have got like 5 different mechanisms of variations, each of varying degree, right? So beyond point mutations, each of varying degree and intensity, you have got multiple mechanisms of variations within the realm of genetics. And the story doesn't end over there. You have got other alternative mechanisms of inheritance as well and variations. You have got epigenetics, you have got methylation, histone modification, non-coding RNA, enhancer promoter interaction, which controls the transcription of certain genes, and the inheritance of microbiome and symbionts, you name it, and you have got parallel mechanisms. So what should happen now is this: you can think, if you can actually, if you have got a modeling mind, you should be able to see that you should have this equation 4.1 defined at the level of each mode of mechanism. So that would be the modern version of the Price's equation, which is defined at the level of different modes of variation and inheritance. And those different modes of variation and inheritance can model changes that vary at different scales and at different time scales. The epigenetic changes can proceed very fast. So you have got like parallel equations changing the characteristics and traits of the population from generation to generation at different speed and with different intensity. So you would probably fill a page of having these 10 kinds of equations, basically the same abstract version mathematically of Price's equation, but each modeling the idiosyncrasies of that particular mode of variation and inheritance. So this is what I would expect from the modeling experts and the researchers who work in modeling to probably step out of genetics. They can actually present and extend a model, and I actually know one of those mathematicians, know in the sense that I have read a book from one of the modern evolutionary, like the extended evolutionary synthesis proponents, that mathematician who is modeling, he was actually presenting strange kind of models even beyond these multiple modes of inheritance. I think his name is Marcus Feldman, okay, if you want to look him up. So there are people who are working on new mathematical models for this extended evolutionary synthesis. But my starting point conceptually, if I want to start small, would to have Price's equation for epigenetics, Price's equation for other non-genetic inheritance, Price's equation for alternative splicing, Price's equation for point mutation, Price's equation for frame shift, Price's equation for enhancer promoter interaction, so on and so forth. And you will see that each one of those equations will govern evolutionary change at different time scales and at different speeds. So that's the deal. So that's our mathematical challenge to the mathematically inclined. And you have to get your hands dirty. It's not that simplistic schoolboy mathematics that Dr. Gelernter tried and Stephen Meyer tried to uh, like uh, like brainwash younger minds that when they think when they encounter fractions they get impressed. That's their upper limit. That's their bar. If you really want to get into some serious mathematics, that's the level of mathematics. And look what the conclusion is. These mathematicians are saying that this equation spells out the return of the original Darwinian logic.
So, guys, this is what we wanted to explain to you today. And I told you that this is not over. We are going to do one more episode that is going to be hopefully next Friday. And that's it. Spencer, do you want to add any concluding remarks before we go?
I know very well. I think it's been like almost 2 hours now. So we
One and a
can
half hours.
1.5 hours. So, but I think we have got other material. Whenever we plan our next episode, we can discuss a number of other. Uh, so I can give you a kind of teaser for what's coming potentially in episode number 3 of this
Four.
series. Uh, yeah,
The three
four.
for this one. Three
Yeah,
for this
three.
one.
Yeah, 3 for this particular, but 4 for the channel.
Mhm.
That would be the sources of innovation, how novelty and innovation evolves, uh, some references and review of papers about de novo genes. We can talk about that. We can talk about an interesting take of speciation as a phenotype and how epigenetic mechanisms can explain the process of speciation as well. So we can point you to some interesting literature on that as well. We can also go through some of the papers of transgenerational or multi-generational inheritance of microbiome and symbionts, which can non-trivially affect multiple generations, not necessarily just 3 generations, not necessarily limited to the grandmother thing that we have covered many times before. Of course, that's very popular. Uh, Changes up to 3 generations, but changes that persist much beyond 3 generations, and what role microbiome, inherited microbiome, plays in the evolution of the host. So all of those things we can cover next time, and we can also cover some of the fundamental sources of mutations. Like even then, why is it the case that the mutation occurs in the first place? And you have to go down to the level of quantum mechanics uh, to understand what happens to the hydrogen atoms in each of these bases and how that changes its position, and as a result, the a different base gets paired with it, and that's a point mutation. So even what is our best understanding of point mutations in terms of quantum mechanics, that's called quantum fibrillation. So we will talk about that. So we will go down to that level, which I don't think so Gelernter and folks have dreamt about.
Yeah.
That what's
But
the
that's
lower.
that's all there in a basic book on microbiology.
Yeah.
That's in the books.
Okay, very well. So that's what we are looking forward to. So stay tuned for the next episodes as well.
Okay, guys. So this is it for today, and hopefully we'll see you next Friday. And let's move on with this. And hopefully you enjoyed today's episode. And we would like you to watch the first part of this that we did uh, just the Saturday before the last one. Sorry, the Friday before the last one. So it happened 2 weeks ago. So I would want you to watch that one as well. and then watch this one, and hopefully we'll bring you the next one soon enough. So thank you very much, and take care, and good night from me and Substantia, and we will see you
Take
next
care.
time.
Good night. Have a good weekend. Bye.